Again, for today's hands-on session, will also be available through Zoom, and today we will be later on analyzing the genome scale metabolic model to identify targets for butanol production in E. coli. So you will be working hands-on with a genome scale model of E. coli and how to analyze it to find targets. So I guess we'll start now. Good morning, all the participants. It's my pleasure today to introduce Dr. Anu Raghunathan, who is currently a senior principal scientist in the Chemical Engineering Division at the CSIR National Chemical Laboratory in Pune. Before that, she was working as a faculty in infectious diseases, working on systems biology of intracellular pathogens at Mount Sinai School of Medicine in New York. Her current research group, the Metabolic Inquiry and Cellular Engineering, in short form MICE group, uses systems approaches, both experimental and computational, for studying biological cells. Research problems include application of metabolic engineering and systems biology approaches to design cell factories for value-added drug products and bioplastics, and understanding drug resistance in glioblastoma and microbial pathogens, constraints-based flux analysis for context-specific models. As an activity of the Pune Knowledge Cluster (EKC), MICE Lab has been working on integrated approaches to COVID epidemic outbreak data analysis, transforming renewable resources like starch into value-added chemicals like lactic acid and diacetyl in her PhD doctoral thesis. Using microbial fermentation and enzymology was the start of a career in metabolic engineering at IIT Bombay for Dr. Raghunathan. Working with pioneer in bioethanol production, Dr. Lonnie Ingram at the University of Florida, and with Bernhard Paulson, a metabolic modeling and systems biology proponent at the University of California at San Diego, developed a flair for incisive, holistic, and systemic thinking. Her previous work on genetic basis of adaptive evolution on suboptimal growth rates was cited by NOVA as one of the top 10 advances in evolution of the previous decade. She is an associate editor for Biosystems, which is an Elsevier publication, an active board member of the International Study Group for Systems Biology, and the founding member of the Bioengineering Society of India. She is also an active member of the PKC, an intra-institutional body sanctioned by the Office of Principal Scientific Advisor to Government of India. Her talk today is titled Exploring Synergies Between Synthetic Biology, Systems Biology, and Metabolic Engineering for Rational Strain Designs and Pathway Engineering. So without further delay, I hand over the system to Dr. Raghunathan for her enlightening talk.
Thank you so much for the wonderful introduction. I would like to, at the start, thank the organizers, about Shams, Shirish, for writing a lot of emails to me. I'm sorry I wasn't very prompt, but I'm happy to be here at this forum. I am a little groggy because I have taken a few medicines. So if you have any clarifications needed, please feel free to stop me and ask me any questions. You can always feel free to talk to me afterwards and write emails and contact me because I'm right here at NCL in Pune. So as mentioned, the title of my talk, which is quite a mouthful, is Exploring Synergies Between Synthetic Biology, Systems Biology, and Metabolic Engineering for Rational Strain Design and Pathway Engineering. And I think it fits in very well. I'm not able to move the slides. Okay, yeah, it fits in very well with the theme of this workshop or this theoretical and practical course at ICGB, the applications of flux analysis and synthetic biology in bioenergy. Although we do not directly work on bioenergy at this point, I think there are a lot of things that one can use, from the methodologies and approaches in this talk and apply it to bioenergy. So I am the principal investigator of the MICE group, or the Metabolic Inquiry and Cellular Engineering group. This is my contact details. The philosophy of our lab is to look at the genotype-phenotype relationship. And in order to do this, ask this very fundamental question, we use a systems biology approach. And systems biology has been defined across the world, across different groups in different ways. But in our group, we like to look at it as understanding heterogeneous data types by using high throughput technologies, and combine experimental and mathematical or computational techniques. So in the end, the philosophy is to use a model-driven or a model-integrated approach to understand the genotype-phenotype relationship, whether it is in a mammalian cell or whether it is in a prokaryotic cell. So some of the model systems, as mentioned in my introduction, that we use look at both drug resistance as well as making value-added products. We use a variety of both mammalian as well as prokaryotic cells. So when I was approached to give this talk, I was really wondering what value-added product I should talk about today. And I have narrowed down, the whole talk to polyhydroxyalkanoids, the class of compounds, and specifically I'm trying to look at polylactic acid as an example, because I think it really fits into the concept of synthetic biology as well. Because earlier I thought this was more a flux balance analysis kind of workshop, but looking at the agenda, I see that it's quite broad. It's much broader than just one area of research. And so I will be talking about PLA, and in the coming slides I will also discuss why PLA fits in and why is PLA so exciting for a synthetic biology researcher. So this is something that I think I've already talked about. We work on all these areas of research. The methods that we use are in this circle. We use constraint-based modeling, we look at metabolism, we look at network reconstruction, and we use a lot of systems biology methods, both experimental and we try to combine and integrate everything and look at what is at that intersection. What lies at that intersection is what we believe will tell us, will help us understand the genotype-phenotype relationship. So moving on to the next slide. So the outline of my talk will be based on the systems biology paradigm. I will talk a little bit about computation that is used in both synthetic biology as well as systems biology. I will look at some of the technologies that one can use to understand, different parts of a system. In this case, I will talk about, since I said I already mentioned, I would be looking at synthesis of polyhydroxyalkanoids. I will talk a little bit about operon synthesis and metabolite measurements, etc. And lastly, but not the least, once you combine computation and technology, what results is understanding the biology, getting new insights in biology. And that insight is in designing a rational strain to make polyhydroxyalkanoate. And eventually, we hope that this will lead to making polyhydroxyalkanoate cell factories. So essentially, this covers the systems biology paradigm, which is both iterative, which uses both experiment and computation, and essentially involves component enumeration, reconstruction, modeling and simulation, hypothesis generation, and biological discovery. And I guess some of you or most of you are probably conversant with some of these item points in this paradigm. So before I dive into polyhydroxyalkanoids, specifically PLA, let me bring your attention to one of my favorite authors, Francis Crick, and his book, What Mad Pursuit, which was essentially a personal view of scientific discovery that he wrote sometime in the 1970s or so, but it is still relevant, very relevant today. So one of the quotations he has in the book is, let me just change, I need to, yeah, because my screen is being masked by a panel, so I'm just going to try and remove that. So what he mentions, one of his very interesting quotes in the book is, almost all aspects of life are engineered at the molecular level, and without understanding molecules, we can only have a very sketchy understanding of life itself. So in my opinion, what the author is trying to refer to are the molecules that are present at the different molecular hierarchies of the cell. That is the DNA, the mRNA, the proteins, and the metabolites. And it is only by understanding these components that one can really understand phenotype or understand the genotype-phenotype relationship. Another interesting quote in the book is that the job for theorists, especially in biology, is to suggest new experiments. That a good theory makes not only good predictions, but surprising predictions that then turn out to be true. And I think this is the agenda of a systems biologist, and this is an agenda of a constraints-based modeler, a flux balance analysis modeler, that essentially you not only look at these interacting components, but you understand the crosstalk between them, because as you know, these models contain all these different layers of molecular hierarchy, and then you are able to predict something that is non-obvious. So why is this useful to us? Why is this concept useful to us? Because we essentially view the cell as a chemical factory, right? So just like a chemical plant has analogous component classes like mixers, controllers, unit operations, many different unit operations, a cell has analogous components like genes, proteins, organelles, some of those molecular hierarchies that I talked about. And the only problem is that the level of control here is much lesser, at a much lesser level of sophistication than in a chemical plant. So one needs to attain that level of fine-tuning if we need to convert a cell into a cell factory. You know, if you really want to move from just looking at the cell as a biological molecule with biosynthesis pathways into a plant like this, we would have to understand how to fine-tune, how to control the different components in the cell, which can be done through both systems and synthetic biology approaches. And they fit in very well into the idea of metabolic engineering, which is essentially making a product, a desirable product from a cell. And if we really step back and think about it, the main objective of the cell is not to make that product. That product is our bioengineering objective. The objective of the cell is to grow, right? That is the evolutionary objective of the cell. So as the cell works to maximize its fitness or growth, as it goes along Darwinian processes of evolution, holds on to those tenets, we want to try and harness its ability to make value-added products. So we need to always look at two competing objectives. When we think of process development, when we think of cells, microbial cells as a cell factory, or even CHO cells, even mammalian cells as cell factories. So some of the model systems that we look at in our lab for metabolic engineering, we've classified them into three different types. One is indigenous to metabolism of E. coli. We love E. coli, so we work with E. coli. Quite recently I've started looking at working on some yeasts. But until now, we've majorly focused on E. coli as our workhorse. So we look at compounds or metabolites that are indigenous to the metabolism of E. coli, like tryptophan, which is an amino acid. We look at some compounds that are non-native to its metabolism, like for example, violacin, which is the smallest indular carbazole. It is formed by a microorganism called Chromobacterium violacum. Violacin is a purple colored pigment, and it is also an antifungal and an anti-cancer agent as well. It is not yet approved by the FDA. We also looked at bioplastics, polyhydroxyalkanoids, and polyhydroxybutyrate, polylactic acid, etc. Now, the third category, which I find very exciting and very relevant to this workshop, is products that are synthetic to universal metabolism, which truly require the principles of synthetic biology to come in play. Because these compounds are not really present in any organism. So polylactic acid, if you remember your chemistry of 12th standard, is actually a polyhydroxyalkanoate. It is the smallest polyhydroxyalkanoate. For most microbiologists, they might feel that polyhydroxybutyrate is always talked about as the smallest polyhydroxyalkanoate. It is the smallest polyhydroxyalkanoate that is made in microbes, polyhydroxybutyrate. Polylactic acid is not made in microbes because the two genes that are required, one to make the monomer and the other, the polymerase, are both not present in the same system. So hence it is very, hence synthetic biology can be used in a very nice manner to make polylactic acid in a microbial cell. So this just gives you an idea of why polyhydroxyalkanoates are important. As you all know, they are the current alternative to plastic polymers like PET, polyethylene terephthalate. We need to replace a lot of the non-biodegradable, non-biocompostable polymers that we are using every day for everyday activities by biodegradable ones. So hence polyhydroxyalkanoates are a very good replacement. The availability of monomers and its cost also make it sustainable and useful and apt polymer to make. So these are some of the pathways for PHA production. So essentially, there are 3 genes that are required in the native organism, Ralstonia eutropha, PHAA, PHAB, and PHAC, which essentially convert acetyl-CoA from the TCA cycle into aceto-acetyl-CoA and then to 3-hydroxy-acyl-CoA. And depending on which acyl group, which R group is attached, it can be polymerized by the PHAC into a polyhydroxyalkanoate. So the problem with polylactic acid is that you need to make the lactoyl coenzyme A monomer. And if the lactoyl coenzyme A monomer is not formed in large quantities, you will not be able to make PHA. So you will not be able to make PLA. So hence, if we need to make PLA in E. coli, we need to add genes from different organisms. So you have, there is an organism called Acetobacter pastorianus, which has an enzyme called the propionate coenzyme A transferase, or the PCT. And generally in the native organism, it is known to transfer the coenzyme A from propionate to lactoyl. And so it can use lactate as a substrate. The other enzyme that is required, so you need to make the monomer. In order to make a polymer, you need to make the monomers, and you also need a polymerizing enzyme. So the polymerizing enzyme that has been used by us is from the bacterium, Chromobacterium violacum. Chromobacterium has a polyhydroxyalkanoate synthase. And the reason why we decided to use it is that it is known to polymerize small length chain hydroxyalkanoates. So this is also very important when you look at polymers. If you want to make small chain polymers versus medium chain length polymers versus large polymers, what kind of polymerase are you using? So essentially, this is a type 2 synthase for low molecular weight monomers. And that is exactly what we are trying to target in the synthetic biology approach. So why can't we just PCR these genes from the native organisms and put them in E. coli, right? That would be a much easier way, a more traditional way to do it. So one of the reasons to use synthetic biology for this purpose is that we are trying to express two genes, each from two different organisms, in a third organism, E. coli. So, you know, based on your first principles of biology, the DNA is not going to be optimal for transcription and translation in E. coli. You're going to have a lot of multi-organism codon bias, right? Your codon adaptive index, your specific codon frequency are all going to be very different for all the three genes. You probably end up with differential GC content, and you might also have a lot of restriction sites which, you know, would induce host nucleus cleavage in E. coli, right? Because you are pulling out genes from organisms that are very different and putting them together in E. coli. So for all these reasons, you know, it's a good idea to go ahead and optimize the gene sequence for both codon usage, so the codon adaptive index, GC content, the restriction sites, and then make sure that the thermodynamics of hairpin formation is also minimized, because you want to make sure that transcription happens very smoothly in E. coli. So on doing this for, oops, sorry, the slide is a little messed up. On doing this for the two genes, we were able to optimize the codon usage to that of E. coli, and it was around 0.98. Oops. Yeah, so it started off with 0.57, and for our gene that we are introducing into E. coli, the codon adaptive index was 0.9 for PCT. And for PHAC, we were able to only increase the codon adaptive index to 0.88. However, we managed to, you know, reduce the GC content quite a bit. So chromobacterium violation is an organism with very high GC content, and we reduced it by almost 15% so that it matches the E. coli. It is not rejected by E. coli. And so then we went on to use typical, you know, molecular cloning methods, and we were able to create a clone. If you look at the top right-hand corner, that image is a synthetic clone that is uninduced, and the one at the bottom where you see the red dots, it's stained with Nile red, and you can see that there is induction. So indeed there was production or synthesis. I wouldn't say production, because production is used more on a chemical industry scale, but there was synthesis. We were able to transcribe and translate the two genes from the two different organisms into E. coli and successfully, you know, make a polyhydroxy alkanoate. So then we were very excited. We went on to use chemical methods to extract. We grew large batches of these cells and extracted them. And then you can see at the bottom of this round bottom flask, there's a plasticky kind of layer. And that essentially told us that we were on the right path, and it was indeed, you know, we had indeed got some clones for making a polyhydroxyalkanoate or a bioplastic. We went on to run FTIR analysis because we need to understand what kind of polyhydroxyalkanoate we are making. Indeed, we were making polyhydroxyalkanoates, but we were rather disappointed that instead of making polylactic acid, we ended up making more polyhydroxybutyrate. So if you go back to the drawing board, what was the problem was we were making, instead of lactoyl coenzyme A monomer, we ended up making more 3-hydroxy coenzyme A, 3-hydroxybutyryl coenzyme A. And that was induced because we were using E. coli as our system, and E. coli already makes hydroxybutyryl coenzyme A as a part of its metabolism. And so it was probably just, trying to move the CoA, the PCT gene was just trying to move the CoA from one part to the other, from one fatty acid to the other. So then we went on ahead to, quantitate it. We figured out what were the different carbon sources that we could make our PHA on. And we saw that glycerol was the most optimal carbon source that could be used for making polyhydroxyalkanoates, or PHB in this case. And then, so for most, if we want to eventually move any strain from the lab to an industry scale, as probably most of you know, we need to make sure that there are no antibiotic resistance genes in these strains, and we want to minimize the use of plasmids. A genome integrated strain is a much better option so that you don't lose some of the genes for making your product of interest. So our next goal was how do we integrate these genes that now we have put on a plasmid and we are working in E. coli with, and we are sure that it can make, it can grow well, it can make polyhydroxy butyrate. So how do we go ahead and how do we, integrate these genes into the genome so that we can potentially then try to use this on a larger scale, on a more commercial scale. So what was very interesting is, we understood a lot about the biology and the beauty of, codon adaptation and synthetic biology, the use of synthetic biology, and that it's just not old wine in new bottle. It really has a lot of implication in metabolic engineering and cell design. So in the process of developing these plasmids to make polyhydroxyalkanoate, we had two parallel approaches. I just mentioned to you that we optimized the gene sequence, and then we added those, we created a plasmid with those genes, and we were able to see polyhydroxyalkanoate formation. We also created on the side another plasmid that had the original native genes. So we didn't change any of the gene sequence. It was the PCT gene from Acinetobacter pastörianus, and the polymerase, the PHA polymerase from Chromobacterium violation. So we took the sequences as such, and then we created a plasmid for that strain as well. And we tried to integrate both these type of genes into the genome of E. coli. And for anyone who has worked on genome integration, you know that it's not a very simple process, it's a much difficult process. But what we saw is that it was relatively quite easy for us to introduce the genes that were optimized for the E. coli genome, for transcription and translation in E. coli genome into the E. coli genome, as opposed to the genes from the native organisms. So this slide here showcases 4 images, and the top images is the E. coli, where we tried to introduce the native genes, and the bottom panel is where we try to introduce the synthetic genes. And as you see, the lower panel shows a lot of fluorescence, 9 red fluorescence. Excuse me. And that's because we were successful at introducing these genes into the genome. So, excuse me. Yeah, so as I told you, we started off wanting to make polylactic acid, right? But when we did our experiments on the drawing board, it seemed like all was right, and we should be able to make theoretically polylactic acid. But when we went and checked our NMR, our FTIR results, etc., I haven't shown the NMR here today, we saw that we were making polyhydroxybutyrate. So what do you think went wrong, right? That means there is some mechanism, there's some part of metabolism that we did not consider, and so we were not able to make lactoyl-CoA. So that means lactic acid is not picking up the coenzyme. So there is an equilibrium, a disequilibrium between the 3-hydroxybutyrate, right, that exists and lactic acid in the cell. And even after we add these genes, the cell prefers to transfer the coenzyme A onto 3-hydroxybutyrate as opposed to lactic acid. So in order to understand what could be happening inside the cell and how do we increase our lactoyl coenzyme A concentrations, we did go ahead and do some measurements for lactoyl coenzyme A, and we saw that there was a very small amount. It was really in picomoles. So it was not enough for us to see polylactic acid being formed. But we knew that small amounts of lactoyl coenzyme A are being formed in the cell. So we decided that we need to use, our mathematical models, and hence this next part of the talk will be more pedagogical, where I will talk about, how we reconstruct our system and what kind of modeling, constraints-based modeling methods do we apply. So this kind of modeling has also been called as two-dimensional annotation of the genome. And that's because we start with the original genome sequence of the organism. So, I just heard, was it Shams who introduced me or Shirish? So I heard them say that this afternoon you are going to have a session where you will use the genome scale E. coli model, right? So assume that you are starting with a genome sequence, say the E. coli model as an example. And this is a toy genome sequence. Let's assume that we have 3 open reading frames, the green, the red, and the orange. Now, these can code for 3 different types of genes. For example, the green can code for a green circle, which can be a regulatory protein, which can bind to the DNA and either transcribe or inhibit, its expression. The red ORF can code for this triangular protein, the red triangular protein, and the orange ORF for a orange square. And these can come together to form a protein complex that can eventually, say, catalyze an isomerization reaction from A to B. Okay. Now, there are many ways of representing this interaction. What we prefer in our lab is matrix formalism. And essentially, if you look at this matrix on the right side, each row represents each component and each column represents a link. So if you look at the first 3 columns here, the first 3 rows, beg your pardon, it's the green circle, right? The regulatory protein, the DNA, the purple, the lavender bar, and the DNA protein complex, the combination of both the circle and the bar. The protein, and this represents the DNA protein interaction. If you look at the next 3 rows, you see the triangle, the square, and a combination of the triangle and the square. So this represents the 2 individual proteins, the triangle and the orange square, and the protein complex, the triangle square combination. So these 3 rows represent the protein-protein interaction. The last 2 rows are much simpler to understand. They are metabolites A and B, right? And they represent components of this reaction that is catalyzed by these proteins. So if you look at a simple 3 open reading frame toy genome, the components that you need to represent in our matrix formalism are about 8, right? And the links essentially tell you whether they are being formed or they are being produced. So for example, if you look at the first link here, it says -1, which is basically the stoichiometry of interaction of the DNA and the protein. It uses one circle, one rectangular bar to make one of these complexes, right? Similarly, there's one triangle, one square to make this complex, which is the protein complex. So based on the stoichiometry of this interaction, you would put down those numbers here, and the sign would depend on whether they are being formed or whether they are being produced. So you can very quickly see that a simple genome here, a toy genome with 3 open reading frames, can skyrocket into a much complex matrix formalism, right? And you can also study using this matrix formalism both regulatory interactions, gene product interaction, and effects on other compounds, right? So at the transcriptional level, at the translational level, and also at the metabolism level. So this is a depiction, this is a system annotation of a small genome here on the left with just 3 open reading frames. Now, if you fast forward and think about the E. coli genome, you all know how large the E. coli genome is, right? So the E. coli genome has 4,400, approximately 4,400 genes. So imagine with just 3 open reading frames, if I get a 8 by 3 matrix, if I have 4,400 genes in my organism, it's going to be really large, right? But what is the advantage of converting a one-dimensional, a primary annotation of a genome into a matrix formalism or a 2-dimensional annotation of this genome? So the advantage is that because it's a matrix, right, you can do a lot of, you can start computations. You have the tools of linear algebra, of convex analysis, all open up to you. And you can use all of that to probe this matrix, okay? So this was very conceptual, right? This was a toy modeling, this was very conceptual. Let me, since I think a lot of you must be biologists or biochemists or life scientists, let me take you to a more familiar example here, right? So these 10 reactions over here on the top left panel, these indicate glycolysis, okay? The first reaction here is hexokinase, right? Hexokinase reaction is, the hexokinase protein is coded by, or the enzyme is coded by the gene GLK, and the reaction that it catalyzes is glucose plus ATP giving G6P plus ADP plus H, right? This is a reaction that you have studied right from your 12th standard, I think, or 10th standard. So if you look at this matrix at the, bottom over here, the bottom left, and just focus on the first 5 rows. I don't know how clear it is on the slide that is being projected there, but if you can see it, the first 5 rows are the reactants and products of this reaction: ATP, glucose, ADP, G6P, and H. And if you look at the grayed part, the numbers indicate the stoichiometric coefficients. Since only one molecule of each interacts with each other in this reaction, we have 1 as the numerical coefficient. Since glucose and ATP are the reactants, we have a minus sign, they are being consumed and they make these products. So ADP, G6P and H have a positive sign, okay? So now each reaction here, so this is the first reaction of glycolysis represented as this, in this red bar, right? Hexokinase, and this vertical red bar here is the reaction vector, right? It's the stoichiometric reaction vector for this hexokinase reaction that is mentioned here. Now, if I look at my 9th reaction here, enolase, right? Oops, so enolase is 2PG giving H2O plus PEP, right? And if you look at my last but one column here, in the matrix, if you look at the last last 4 rows of my matrix, I have 2 Pg, PEP, and H2O, and here I have again the stoichiometric coefficients as my numerical coefficients, and the sign depicts whether they are reactants or products. Now this again is a stoichiometric reaction vector for this reaction ENO. So essentially this matrix that I represent here is a combination of a lot of column vectors that represent reaction vectors, okay? And if I look at, so you can essentially connect the biochemistry that is happening in the cell with mathematics, right, so that you can create this matrix. Now what have I shown on the right hand side, right? So if any of you are familiar with the E. coli genome, the genes are depicted as B numbers. So here I have the B number B2779, which represents enolase, and enolase makes the transcript ENO, which is given in pink square here, and it gives the protein ENO in this oval, the beige oval, which catalyzes the reaction capital ENO in blue, right? We have seen that this capital ENO is depicted here in the red horizontal bar on the left side, 2Pg giving H2O plus PEP. I'm sorry, I forgot to mark it on my matrix here. It is the last, it is the second last column in this matrix, if you can see my arrow, okay? So this is the mathematical formalism, this is the biochemistry, the biochemical reaction, and this is the genetics behind it. This is what allows the whole thing to happen in the cell, right? So in our genome scale models, we have all these 3 layers built in. We have a layer, we have used Boolean logic to represent gene, protein and reaction relationships. We use stoichiometry and chemistry to represent the biochemistry. We use mathematics to represent the stoichiometry of that reaction as a reaction vector, which eventually, when put together for, you know, the 4,400 reactions of E. coli the 2,000 odd reactions of E. coli will form a matrix. Now you may ask this question, enolase seems to be a very simple gene. It's a single gene, single protein, a monogenic relationship, right? But what happens when there are lysozymes? What happens when there are protein complexes? If you remember your biochemistry, a succinate dehydrogenase, which is a part of, which is a part of the TCA cycle, has 4 complexes, has 4 proteins that come together to form a complex, right? SDHA, SDHB, SDHC and SDHD. So all of these come together and form a protein complex, and we can represent this using Boolean logic. So as you know, Boolean logic uses the AND or logic, right? So you can use this logic to depict these gene connections. So for example, here if you look at pyruvate kinase, right, which is on the right hand side next to ENO, there are 2 B numbers, there are 2 genes that make 2 transcripts, PYKF and PYKA. They both make their individual proteins, PYKF and PYKA, but either one of them can catalyze the reaction pyruvate kinase, that is ADP plus PP giving ATP plus pyruvate, right? So we have these OR relationships built into the model. Similarly, if you look at the gap reaction below, the gap dehydrogenase reaction, it looks even more complex, right? You have a straight line, one monogenic relationship here on the left side, and you have an AND relationship on the right side, and so you make 2 proteins, gap A and gap C, but either one of them can catalyze the gap G reaction. So the beauty of these stoichiometric network reconstructions, these genome scale models as we use it today, are that we can depict all this genetics in one layer. We have the biochemistry in another layer, and then the final outermost layer is the mathematics or the matrix. And the reason why we play around with the matrix is that the matrix, as I mentioned a few minutes ago, allows us to use the tools of linear algebra, linear programming, and convex analysis. And I think Dipali and Matthias and David Fell yesterday probably touched upon some of those basic concepts, right? From the kernel matrix and so on and so forth. So on the left hand side here, you have a biochemical network that represents the E. coli metabolism. It has amino acid metabolism, central metabolism, nucleotide metabolism, you name it, it's there in it. And all of these are represented as a matrix that is depicted on the right hand side. Okay, so the metabolites are the rows, the reactions are represented by columns. The elements of this matrix, I'll revise this concept again, are the numerical coefficients of the stoichiometry. The sign is the directionality of the reaction. Is it a product? Is it a reactant? Okay. And this is what forms the stoichiometric matrix, the S matrix or the N matrix, as David must have mentioned yesterday, or Dipali might have mentioned as well. Right? So now once you have this network reconstruction, what do you do with it? So in our lab, what we do is we use what is a philosophy called the constraints-based modeling philosophy. And the basic premise of this philosophy is that if you eliminate solutions of behaviors that cannot be exhibited by this network, right? And you eliminate them by applying physicochemical principles. You don't just remove them willy-nilly, right? You know that there are certain things that a cell can do. Like for example, you know that if you get a certain salary, right, you cannot spend more than that salary, right? Of course, these days you can with your credit card, but if you have to use only cash, you cannot spend more than that. So there are some thermodynamic constraints that govern, cell behavior. There are some physicochemical constraints that govern cell behavior. So we assume that if we eliminate everything that a cell cannot do, whatever remains is what the cell can do. So let me give you a simple example. Again, we go back to a toy network. Here, earlier we had 3 open reading frames. Here I have 3 reaction fluxes, A, B and C. Let's assume that this is the unbounded space that the cell can do anything. Okay, this particular organism has only 3 fluxes and it can do pretty much anything that it wants. And this is the space, so the coordinates are really large, right? Now successively in this cell, I apply physicochemical constraints like stoichiometry, like I mentioned before, limited thermodynamics, like even if I just apply reversibility principles, right? Some reactions are reversible, some reactions are irreversible, right? So I use those limited thermodynamics in. If I look at enzyme capacity or Vmaxes of the enzyme, if I look at genetic regulation, if I have more experimental data and look at concentrations and kinetics, right? If I have kinetic information. If I apply all these constraints or some of these constraints successively to this unbounded space, I end up with a more narrow space. And this narrower space is called the solution space. Okay? And then I assume that if I have eliminated what the bacteria cannot do or what the organism cannot do, that the phenotypes that I observe, right? Say I give E. coli glucose and it grows on glucose with a certain growth rate. And then I give it a compound X that it doesn't grow on. So that means that the compound, this phenotype that E. coli grows on glucose will lie inside this polytope. But the phenotype that it does not utilize compound X will lie outside. That is the unbounded space, right? That is the space all outside. So from so much of huge space, unbounded space, I have narrowed down to a solution space, which will help us as researchers, right, to understand the biology, to understand genotype-phenotype relationship, to understand how cells behave, to understand metabolism, and if you understood from the previous slides, you can also understand enzyme kinetics, right? You can understand enzyme capacity, so it allows you to, understand the organism very well. And how do you create constraints, right? You can apply any constraint here. And as I've mentioned here, I have only mentioned about 6 constraints. So if you do experiments, any experimental data can be used as a constraint. Okay, if you do gene expression, those gene expression experiments can be used as constraints. If you look at metabolite profiling, you can use metabolite profiling as constraints. You can look at the exometabolome, you can calculate uptake rates, and then you can use those as constraints. So essentially, we have the gamut of experimental science open to us to develop constraints that can help us redefine our solution space, narrow our solution space so that we can understand specific phenotypes. Of course, we might have to develop methods to, apply these constraints. So this whole philosophy of constraints-based modeling has been very neatly captured in this saying or this quote from Sherlock Holmes, and I have taken this from one of my colleagues in the Paulson lab, this quote is from a study in Scarlet, and Sherlock Holmes tells Watson, "How often have I said to you, when you have eliminated the impossible, whatever remains, however improbable, must be the truth." So I'm sure that all of you in your experiments have always come up with something where you're unsure, right? So constraints-based modeling tries to remove that unsurety because you have removed whatever is not possible. So whether you believe it or not, whatever you get is closer to the truth, if not the complete truth. And there have been many constraints-based methods that have been developed, over the last decade or more than a decade. Many of them have been used. I think you will be using many of them in your workshop and your hands-on tutorials that are going on for this workshop as well. Some of the most popular are flux balance analysis, flux variability analysis, robustness, phase planes, etc. But there are a lot of new ones that are coming up. And basically these are methods that are developed to analyze that solution space, this polytope that I talked about, so that you can, understand or compute phenotypes of the cell, understand cellular behavior, and these solution spaces are generally defined by both biological and physical constraints, and so applying them allows you to get closer to developing a virtual cell. So let me just, give an example of one of the constraints-based methods here. One of these constraints-based methods is the phenotypic phase plane analysis. And a phenotypic phase plane or a phase plane is a two-dimensional region. It is spanned by two metabolic fluxes, typically uptake rates. And what are uptake rates? So imagine that you put E. coli into a flask and you are feeding it with media, right? So, and this media has glucose. You also have oxygen that surrounds the cell, so you have an aerobic growth. So your one uptake rate could be your glucose uptake rate, and your other uptake rate could be your oxygen uptake rate. Okay? So a phenotypic phase plane essentially is defined as spanned by two metabolic fluxes. It's typically defined by uptake rates, and the lines in between demarcate phases of constant shadow price. So I think yesterday you must have heard, I'm sure definitely, David Feld would have given you some introduction into optimization, linear programming and optimization, or Dipali might have on the first day given you an introduction to that. So you understand that there's linear programming that you're applying, and when you do linear programming, when you do sensitivity analysis, there is a shadow price and there's a reduced cost. So if you look at the shadow prices and you demarcate lines, you draw lines on the solution to indicate constant shadow prices. Then you can divide your solution space, you can divide this space, this two-dimensional region which is spanned by these two metabolite fluxes into different regions which tell you what metabolite will be secreted in which phase. So for example, here if I have metabolite flux A as my x-axis and metabolite B as my y-axis, and I have demarcated, these white lines demarcate constant shadow prices, right? So the topmost here is an infeasible steady state. The bottommost here is also an infeasible steady state. But the ones in between here, there's a certain shadow price, a specific shadow price A, an integer A, and that defines a metabolic phenotype A. So this could be the phenotype. Like for example, when you're growing cells in glucose, if it is completely aerobic, you will get CO2 and H2O, right? All the glucose will be oxidized to CO2 and H2O. So that could be defined by your metabolic phenotype A. And your metabolic phenotype B could be when oxygen is limited and you are making some side product like lactic acid or succinate or something like that. Okay, so by definition, metabolic pathway utilization is different in each region of this phase plane. Okay, there is a line of optimality that can be defined on this phase plane, and that corresponds to condition that both your metabolic flux A and B, they optimize your third component, that is your biomass E. Okay. So there are different characteristics of phase planes, and essentially some of the characteristics are that there are infeasible regions. That means the fluxes don't balance. So you know that the whole point of flux balance analysis is to balance the fluxes, right? S dot V equal to 0 or N dot V equal to 0. So there are regions of single substrate limitations that you can find. So where your shadow price alpha is equal to 0 or infinity. There are regions of dual substrate limitations that you can find where alpha is less than 0. There are some futile regions. There are some futile cycles that may happen in the cell. There your alpha is greater than 0. There are isoclines, okay, like constant height in topography maps, if you remember your geography. And there is a line of optimality which corresponds to the maximal biomass yield, okay. That is the ground cells per millimole of the carbon source that is utilized. And you will generally find this by fixing your carbon uptake rate and optimizing for biomass using FPA, and this will give you that 1 point on your line of optimality. So these are some, so this is like a representation of what are the lines with constant objective values which probably you will study in your, in the workshop when you do more hands-on. And your line of optimality here, this is the same figure I showed you first to define what is a phase plane. A phase plane comes from simple mathematical analysis when we are applying it to biology over here. This line of optimality here defines the maximum yield, that is the YXS, okay, which you might be familiar with, right? Because you have glucose and you're making a product, say lactate, and you have utilized all your substrate, then whatever biomass you get at the end is your YXS, right? So it's a YX0, XT minus X0 divided by YS minus ST. So this is your line of optimality. So let me bring you to something more exciting, which is quite theoretical, right? So one of the earliest experiments one of my former colleagues did was to test the optimal growth hypothesis. So what was essentially done was that E. coli was grown in 2 grams per liter of malate, okay, and in minimal media. And the malate uptake rate and the oxygen uptake rate were calculated and they were determined experimentally. Okay, so on the left panel here you see a 2D projection of the phase plane. So the phase plane is generally a plane, right? So it's 3-dimensional. Here this is a 2-dimensional representation of it. And you can see that the x-axis is the malate uptake rate. It is shown as MUR or flux B. The phase and the oxygen uptake rate is given in the y-axis. The phase plane is divided into 4 regions. Phase 1 is characterized by metabolic futile cycles, like I mentioned in the the toy example. Whereas phase 2 is characterized by acetate overflow metabolism. Okay, this is over here. Metabolic phenotype A, acetate. The purple line is pointing towards it. The line of optimality is shown in red, which basically separates your phase 1 and phase 2, and this line of optimality represents the highest growth rate. The line of optimality, essentially maximizes the biomass yield. So our hypothesis was that cells prefer to grow on the line of optimality where they produce the highest growth. Okay. So the data points that are shown here represent malate concentrations that range from 0.25 gram per liter to 3 gram per liter. I'm just looking at my notes to make sure, because it's an old experiment that was done in 2000. The two data points in blue that you see, they represent, oh yeah, so I have to move on to my next way. This is the plane, right? So this is a 2D representation on the left of the 3D phase plane. So in order to understand the shadow prices, these are the isoclines, these black lines are the isoclines. This middle line here that you see here, which has LO depicted on it, is the line of optimality. This is the line of maximal growth. Your phase 1 here is the infeasible steady state. Your phase 2 is where you make more acetate. Your phase 3 here, as shown in the 2D projection, is where you make more formate. Okay. So here you see that the XN, lot of the points, the growth rates, right? If you look at this 3D representation, you see that there are a lot of points that are present on this line of optimality. So they essentially show the growth of E. coli on malate. So, you know, it seems that growth optimality principles on malate are followed by E. coli. So E. coli grows optimally on malate. But how about other substrates, right? So do all substrates show optimality for growth? So then this is so this is for, this data is for glycerol over here, and as you can see here, the white dots, I apologize for the blurry slide. So these white dots here represent the growth of E. coli on different concentrations of glycerol, and you can see that they lie far away from the line of optimality on the top graph, right? So that shows that E. coli grows suboptimally on glycerol. Now, the figure here at the bottom essentially represents all these white dots on this line of optimality, right? So what has happened between the top and the bottom graph here? What does this arrow depict? So this arrow essentially was, the strategy of adaptive laboratory evolutions, which I think if most of you who are attending this seminar, or most of you are attending this workshop or working in this area, are very familiar with. ALEs have become a very common tool, a very useful tool for metabolic engineering purposes. If you backtrack to a few slides ago, where we talked about making a strain of E. coli integrated in its genome with a propionyl coenzyme A transferase to make, more coenzyme A, lactoyl coenzyme A or a butyryl coenzyme A, and a polymerase enzyme. What we did was we decided to subject that particular strain to different selection pressures and to see whether we could improve either the growth or the yield of the polyhydroxyalkanoate that we were looking for. So one of the other biggest questions, I think, for people who are interested in flux balance analysis or metabolic modeling is that is it really right to maximize for growth rate, or should we maximize for growth yield, right? And the answer to that question can only come from experiments. The answer for that question can come from biology. Does the cell optimize its rate or its yield, right? What is fitness? What is evolution? Those are the philosophical questions, or those are the biological questions one should ask the cell in order to answer a question that a lot of metabolic modelers have been asking over the last several decades, maybe half a century, even before flux balance analysis became popular in biochemical biochemical theory? That is a big question. So that was one thing that we wanted to explore, to see whether since now we have an E. coli that makes a particular compound, PHA, which is not in its general repertoire, if we were to evolve it, if we were to passage it every day on media, would we end up with a strain that has maximized its growth, or would we end up with a strain that has maximized its product formation, right? As a bioengineer, I would want it to maximize its product, right? But as a cell trying to survive, it might probably not. So that was when our story began, our day 0, where these were the initial growth rates and PHB production rates for us, which were very minimal. As you can see, we had very low quantities of PHB that we measured out. We know that we are making them because we can see this fluorescence in the tube. And then we subjected to evolutionary pressures. We used two evolutionary pressures here. We used glycerol and we used arabinose. Glycerol because, I told you earlier, we did a substrate optimization and we found that glycerol was the most optimal substrate. And arabinose because it was one of the inducers that we thought, since we were introducing, we wanted to make sure that arabinose, the PBAD promoter, had not entered the E. coli genome. So these are this is very preliminary data. We have much more robust data now, but unfortunately I won't present it today because I was, because I got tested positive for COVID. I haven't had time to compile all the data in here, but probably in the next presentation, the next talk I give, we'll probably have the updated data set. We've evolved them on many more substrates now, on acetate, on lactate, because we believe that only changing the substrate selection pressures will allow us to change our monomer that we are forming. And one of our main excitements is to make our lactoyl coenzyme a monomer, so we can make polylactic acid in larger amounts. So this is just a snapshot of our evolution. We see that growth, as we all know, growth increases with evolutions. We also see that the rate of PHB production did increase with time. So this is data that's sort of fresh out of the oven. So in conclusion, synthetic and systems biology approaches are a common denominator for metabolic engineering, and they allow for scalable methods to increase yields of any desirable metabolite. In our case, I've shown from the previous slide that we can increase the yield of PHB. You can use modular strategies that are scalable to multiple monomers, right? Because the idea is just not to make PHB. We would like it to be a scalable platform to make PLA, to make PHB, to make PHBV, to make the larger, the medium chain length and the higher chain length polyhydroxyalkanoids. Adaptive laboratory evolutions have become a mainstay, and they are really useful, as seen in the previous slide, and as seen in many other studies that have been published so far. Constraints-based modeling, reconstruction and analysis is extensive and is very critical to understand bottlenecks, right? We've also understood certain bottlenecks by applying other constraints-based modeling approaches, which I didn't discuss today. We've identified certain genes that need to be knocked out to be able to increase lactate production and to reduce acetate production, and so on and so forth. Phenotypic phase-spin analysis is a wonderful mathematical technique and can give an idea of all the substrate limitations and the impact on growth and product formation. It's a very good idea to look at phenotypic phase-spin analysis with cofactors specifically. I didn't get time today to talk about cofactors, but NADH, NADPH are very critical to not only production of PHA here, but to almost all the compounds I showed in our, what our mice lab works on, and the larger gamut of compounds as well. So PHPP analysis really gives you an understanding of how, on one of the axes, you can have your, substrate uptake, right? Your flux A could be your substrate uptake. Your flux B could be your, oxygen uptake. And instead of having your biomass that you're looking at, you can look at cofactor formation. And you can get insights into how cofactors will modulate your product formation. So with that, I would like to thank my Mice Lab group. I have 3 iterations of the Mice Lab. This is my very first Mice Lab. As you can see, I look much younger than I look today. This is my 2nd iteration. I've grown older. And this is my 3rd iteration. We still have a few people missing in the picture because, I have not been able to create I wanted to create one over the last week, but as Murphy's law dictates, I did fall sick and I didn't have time to collect all the pictures and create a nice composite map. But I thank all Mice Lab members over the years for supporting this research and putting up with my idiosyncrasies and talking about constraints-based modeling. I would also like to acknowledge my funding sources, DST, DBT, CSIR, and Gates Foundation. I would like to thank my collaborators at NCL for all my LCMS work. I would like to thank my friends at Oxford Brookes, David Fell and Mark Pullman, for wonderful discussions. I actually was hoping that I could meet them at Delhi this time, but maybe another time. For great discussions with Stefan Schuster about, understanding the biochemical theory, the biochemical system theory. And my collaborator from University of Toronto, Krishna Mahadevan. So I would like to thank all of them and my funding sources. And I will, end with that. Happy to take any questions. And if you need to contact me, if I was not very clear today, I'm sorry. I'm still under the influence of a bunch of drugs. And you can please feel free to write to me or contact me. Some of my students are attending this workshop, so you can, talk to them as well and get my contact information from them. Thanks a lot, and I'll be happy to answer any questions now or later.
Thank you, Dr. Raghunathan, for a very, very interesting talk in covering a variety of topics, both in terms of bioplastic production, phase plane analysis, adaptive laboratory involved evolution, and also in terms of synthetic biology, how, for example, codon optimization, etc., are very much needed to have good expression. So, yeah, we are open for question. Since we are running a little late, then our schedule. So I think what we'll do is we'll take just a couple of questions at this moment, and since we also the next speaker will join it around 11, so we need to have a quick tea break. But anyway, let's have a couple of questions, please. Online participants, do you have any questions? Yeah.
So either everyone understood everything or didn't understand anything I said.
So yeah, we have one question from the audience here.
Hello. Good morning, ma'am. Thanks for the very nice presentation. I have a question that as you tried to engineer the E. coli cells for the production of PLA, you said that the CoA was being transferred to probably PHB and you didn't see any PLA production. So did you try any alternative strategies? Did you try to delete, knock out the PHB to produce so that you can successfully produce PLA?
Yes, so I mean, unfortunately, you need to make 3-hydroxybutyrate for central metabolism of E. coli to keep E. coli happy and grow. So you can't really cut it off completely, but you can change the equilibrium so that you don't have too much of 3-hydroxybutyrate and you have more of lactate, and that can go into the pathways. So what we are currently doing is we are trying two-pronged approaches. One is to knock out certain genes so that we can reduce 3-hydroxybutyrate, and also change the substrates that we give. So currently the substrate that we have looked at in detail is glycerol, and glycerol is known to make is known to feed into these pathways and make hydroxybutyrate. So what we are trying to do is we are trying to look at other alternate substrates that can probably increase lactate production, and therefore we will have a transfer of the coenzyme A onto lactate as opposed to and we'll make more lactoyl CoA as opposed to more butyryl CoA. The other strategy, the third strategy we are looking at is, can we put some selection pressures and increase lactoyl coenzyme A formation. So we are looking at 3 alternate strategies. Definitely we have not given up on making PLA, because we have seen that in our NMR, in our LCMS studies, that we have identified small amounts, very small amounts, less than picomolar amounts of lactoyl coenzyme A. So we know that we need to figure out ways that we can increase this particular compound. Yeah.
Thank you.
I hope that answers your question.
Yes, thank you.
So Tanika, can you wait, because there is an online question. So we'll take your. Fabian, do you want to ask online? I mean, in your own voice, or do you want me to speak out your question? Well, I'll speak it out for the interest of time. So his question is, how many cycles of selection normally are required prior to industrial usage? So I think he's talking about ALE. So how many cycles would be needed if you want to make a product at an industrial scale?
So that I think would be product specific. I don't think there is a magic number per se. It also depends on how badly we have treated our industrial strain, right? I mean, if we are going to add, say, 15 genes or say 5 genes to E. coli, then E. coli is already under a lot of stress, so it might need more, generations of evolution before it sort of stabilizes and it becomes comfortable in this new form. So what ALE does is it just helps, it adapts the cellular architecture, both the physiology and the genetics, to get accustomed to the conditions, both the internal conditions, the gene conditions that it has, any knockouts that you've made, any new gene additions that you have made, and it also allows you to adapt for the substrates or the raw materials. So I think if you have a strain that is ready and that makes a certain compound of interest to you, you should passage them, you should go through multiple generations, and you should be saving samples, you should be freezing down these cells, and you should be then testing them out for your product of interest. And generally a good time to stop is when your growth stabilizes or your product stabilizes. So you know that say after 100 generations of evolution, the cells do not increase their growth rate. Like for example in glycerol, when you grow E. coli, when you grow E. coli on glycerol, the initial growth rate is around 0.22 hour inverse in M9 media. When you passage them for about 750 to 1000 generations, the growth rate increases to more than twice to 0.55 hour inverse. Now, from the reason I said when you passage them from 750 to 1000 generations, because after 750 generations, it hits 0.55 and then it remains 0.55 hour inverse for the rest of the time. So we don't really see that there's any change in, that means it has completely adapted to growing on glycerol. Now, if you are making an industrial strain where making high density biomass is not the main objective, but to make a product is the objective, I would suggest that one would have to measure the compound, right? Whatever we are interested in. Say we are interested in making ethanol, then we measure ethanol and see whether ethanol has plateaued. And once it has plateaued, we know there's nothing else that can happen, at least under this certain selection pressure, that will increase your ethanol yield, and then you stop at that point. I hope that answers the question.
So, Sutanuka, last question.
Hello ma'am, very nice talk. So
Hi.
this question is in continuation to the first question, that the PLA versus PHB issue. As you also mentioned that PHB is necessary for the survival of the cell, is it possible to create a conditional mutant of the cell where in conditional mutant?
Can you just repeat the question, the last sentence? Can you create what?
The question is how do you solve the problem of synthesizing PLA, because most of the product is getting skewed towards PHB. So my question is it possible to generate a conditional mutant where in one condition the cell is producing PHB for its survival, and in another condition it is producing PLA, and can this thing be modeled through constraint based modeling?
So I'll answer it in reverse order. I think it's always easier to create models than to actually do experiments. So I'm sure we can create something in a model. But I have a feeling that because, see, this whole metabolism we are trying to tweak, right? If you remember the pathway slide, am I still sharing my slides or did I stop sharing them? So if you remember the initial slides that I showed the pathways, before you enter, just one second, let me just share it. Can you see my screen?
Yes, you can.
Yeah. So if you look at this slide, right, these are the pathways. This is a schematic representation of the pathways for PHA production, and the green part over here is the actual PHA pathway, right? What we are trying to tweak is making this 3-hydroxyacyl-CoA, the last green boxed compound. If you look at the sources of this last green boxed compound, it comes from either fatty acid de novo synthesis on the left side, or it comes from fatty acid beta oxidation. Even if you look at the first box in green, that it comes from PCA. So as you can see, it's a rather tricky problem to make conditional mutants because you will end up losing something, right? Your cells are not going to be happy. So maybe you can make a conditional mutant for as an academic exercise. It might still work out, in my opinion, and I am still learning. But I think if we have to take this to the industrial scale, it has to be a mutant. It has to be a mutant. It has to form more lactoyl coenzyme A. It cannot be a conditional mutant, because then that would create, like, unless in industrial production we are interested in a combination, right? Because there are uses of copolymers, right? But here we are looking only at a case where we want to make a pure homopolymer. So there are, so I think a conditional mutant might be helpful when you want to limit, or you want to control the amount of your second polymer. When you want to make a copolymer, I think it's a great idea. I think a conditional mutant might help. So you can regulate the amount of butyrate coming in. Say I want to make 3 PHBV, butyrate and valerate copolymer. So I think in that case, and I want more of valerate, I think it will definitely help. But I think one of the problems here is because of central metabolism being very rampant, and and fatty acid synthesis being needed here. And currently because we have not really overexpressed anything to make dehydroxybutyryl coenzyme A, that means whatever is excess of what is needed for growth is being diverted here. So the only other way is to divert that 3-hydroxybutyrate to some other pathway probably. Yeah, so I think it's a hard question to answer.
Yeah. All right. So thank you, Dr. Raghunathan, for the lovely talk, and I'm sure with the interaction we had seen that I'm sure there are many more interesting questions that can come up, but in the interest of time we need to move on. And thank you very much for taking time in spite of recovering from COVID once again. And
Thank you so much.
Wish you and your family a very, very quick and complete recovery. And thanks again.
Yeah, thank you so much. Thank the organizers, thank all the audience, and I hope I was clear enough. And please feel free to contact me when I'm better in my senses via email or phone. Thank you. Thanks a lot. Bye.
So, for an announcement for the online audience, so we'll be joining the next presentation at around 11:15 IST, that is 20 minutes from now. So please be online, or you can come join back in another 15-20 minutes. next talk in this series is by Dr. Amit Kosh. Dr. Kosh is an assistant professor of School of Energy Sciences and Engineering with a joint appointment in the P.K. Sinha Center for Bioenergy and Renewable at the Indian Institute of Technology, Kharagpur. He earned his PhD from Indian Institute of Science, Bangalore in 2009. Thereafter, he did his postdoctoral research with Dr. Nathan Price at the University of Illinois at Urbana-Champaign during 2009 to 2011. As a postdoctoral researcher, he worked on metabolic engineering of yeast in the Joint Bioenergy Institute, JBEI, Lawrence Berkeley National Laboratory from 2011 to 2015. His current research interests include genome-scale metabolic modeling and metabolic engineering of yeast for bioproduction, gut microbiome, and community metabolic modeling. He has been a recipient of Faculty Excellence Award 2020 at IIT Kharagpur and Ramalingam Swami Fellowship from 2015 to 21 from Department of Biotechnology, Government of India. So his talk today is titled Metabolic Flux Analysis and Metabolic Engineering for Microbial Production of Biofuels. So I'll hand over to Dr. Kosh to give this presentation. Yes, we can see your slides, Dr. Kosh.
Thank you, Sirish, for nice introduction. Really appreciate for the invitation to give a presentation here. It was a nice workshop, and hope you are enjoying this workshop, which is on metabolic flux analysis and synthetic biology. Is it audible?
It's audible.
Okay, thank you. So, thank you for the introduction, and so far we have seen a very nice response for this workshop. Hope you are enjoying the workshop on flux analysis and synthetic biology. So today I will be talking about metabolic flux analysis. I hope you already listened to few talks on metabolic flux analysis and synthetic biology for microbial production of biofuel. So how we can actually do the engineering, and as well as you can do the metabolic flux analysis, so that you can reduce your effort. Because metabolic engineering is very laborious process, right? It takes a lot of money on also time. So how you can actually reduce the time and can do more effective work. That is the prime objective of my presentation today. So far, I have been working in the metabolic flux analysis and metabolic engineering for production of biofuel or any chemical. So most of the gasoline and chemical, if you see, it is derived from petroleum, raw petroleum. So the raw petroleum, the fossil fuel basically. So this, so most of it goes into gasoline production, which is raw petroleum, and then around 30 to 40% goes into different kind of chemical that is used in our daily life. Okay. So ethanol, you already heard about the ethanol is a renewable fuel, and ethanol is blended with gasoline like petrol, 5 to 10% it is mixed today. What you use in the in your vehicle. So the main idea you know already that if you use biofuel, for example bioethanol, then as you know the plant grows on carbon dioxide. So if you burn bioethanol, then the carbon produced from bioethanol goes into the atmosphere, and those carbons are again utilized by the plant. So there is a carbon neutrality which exists if you use biofuel. In comparison to fossil fuel, if you consume fossil fuel, that means you are releasing the carbon which is stored inside the earth for millions of years. So day by day you are adding carbon in the atmosphere by using gasoline that is available in the petrol pump. And we have 3 generations of biofuel. So the first generation which deal with the food, for example corn, sugarcane, and then second generation biofuel is also known as cellulosic biofuel, which uses lignocellulosic biomass, which is shown over here. You can see the lignocellulosic feedstock. Basically non-food part of the plant is used to produce bioethanol, whereas in USA they use corn for production of bioethanol, and in Brazil we have seen that they use the sugarcane for production of bioethanol. And the third generation biofuel deal with the algal biofuel, which doesn't require much land for its growth. So we'll discuss about how you can actually produce these biofuel from lignocellulosic biomass. The lignocellulosic biomass which absorbs sunlight, and then the lignocellulosic biomass has to be decomposed so that you can get the monomer sugar, and this monomer sugar is eaten up by the microbes. So this is the overall pipeline for the biorefinery we have today. Slowly it is growing, where you have to engineer the microbes. And engineering microbe, how you can engineer so that you can have a desired molecule. It can be a biofuel molecule or a different chemical today. Many research groups are trying, many industries are involved where microbes are engineered to produce your desired molecule. So to engineer the microbes, you can, many people follow this design, build, test, run. So first you design the metabolic pathway that you want to build in the host organism. For example, we have shown here yeast. Yeast is designed in such a way that your desired molecule you can make inside the cell, and for that you can use genome engineering tool like CRISPR-Cas9, and then you can build the pathway, and then you have to test those pathway using high throughput screening, or you can do some measurement using DC and try to understand the production of the molecule. And also you can do the metabolic flux analysis to understand how the metabolic pathways are affected because of the engineering. So now this engineering is the addition of metabolic pathway that can come from plant. So you want to add a gene from a plant or a mammalian cell into this host organism. So there are mainly 2 popular hosts, E. coli and yeast. Yeast basically Saccharomyces cerevisiae. So that can be engineered so that for the production of biofuel. So we'll learn how the metabolic flux analysis work and how engineering is done, because the metabolic engineering is a very laborious and time-consuming process, and it involves a lot of money. For example, some of the products which are in the market, for example, artemisinin, which has been the project ran for 10 years with a cost of $50 million. And it was successfully engineered, the Saccharomyces cerevisiae, to produce this drug for malaria, and it was a successful project. And then another project which was run by DuPont, which produces carbon fibers, and this project cost around $130 million, and it went for 15 years. So the metabolic engineering, what we see that it takes a lot of money, and as well as is very time intensive. So we invest a lot of time as well to get the engineer strength so that it can produce your desired product. So to do this time consuming and expensive process, we will talk about some of the tools which can enhance your engineering. So it can do in a rational way so that you can quickly get your product in a shorter time. So first we'll discuss about the systems biology, which is the topic in this workshop. So the systems biology, how you want to map the genome into. So generally the central dogma of biology, as you know, that you have DNA, and then you have the RNA from the DNA, and then protein. So mostly people stop here with the protein itself, but in systems biology we go from mainly from we map the protein into reaction. So from DNA, from genotype to phenotype basically. So if you need to know the mapping between the gene which catalyze reaction. So this mapping you need to understand in order to build the metabolic network or genome scale metabolic network, which you have already studied in this workshop. So I'll just extend a little bit from here. So the flux analysis basically you should know that what is flux. So flux is not a metabolite concentration. So that is the rate of molecule going through a reaction. Suppose I consider a reaction X, then the number of molecules going per unit time through a reaction that is given by a unit millimole per gram dry solvent per hour. So that the amount of the rate at which this molecule A is converted into molecule B. So to calculate the flux, there is no equipment or any instrument which exists today which can give you directly the experimental value, the rate at which, and this you want to do at the genome scale level. You might get flux for 2, 3 reactions, maybe 10 reactions you will using Michelson mental kinetics, but if you do at the genome scale level in a high throughput fashion, but high throughput technology like genomics, transcriptomics, proteomics, and the metabolomics, they are done in a high throughput way. So fluxes, to measure the fluxes, we need to do some metabolic modeling. So for that you need a metabolic network. Before you calculate the flux, you should have a network. And then if you can calculate the metabolic fluxes for the entire network or at the genome scale network, you can calculate the fluxes. So similar to genomics, transcriptomics, proteomics, which you are already familiar by measure the transcriptome of entire cell, proteome of entire cell, metabolome of entire cell. Similarly, fluxes are also can be estimated for the entire cell. So with this idea, we can also draw a metabolic flux profile like this. So this is just a central metabolic network where you can see the carbon glucose is entering, and then some of the carbon goes in PPB pathway, some carbon goes into glycolysis, and then it ends up with TCA cycle. And this way you can draw how the carbons are flowing in different metabolic pathways. To calculate the fluxes, another terminology is known as flux balance analysis. So flux balance analysis, which calculates the metabolic fluxes quite easily without much. You don't have to do any experiment to calculate the metabolic fluxes. Just we need to have a metabolic model. So here you can use an annotated genome and try to understand how many reactions this genome have. And then using this reaction, one can construct this stoichiometric matrix, which is basically the columns are the reaction and the rows are the metabolite, and the element inside the matrix are either -1, +1. That means -1 means that metabolite is reaction, and +1 means the metabolite is product. And if you have a stoichiometric matrix, then your job is easy. You can go for optimization, because fluxes can be estimated using this kind of stoichiometric matrix, and then have the boundary condition, and then you go for maximization of the growth rate. So this is very well understood in terms of microbes, where microbes, you know that the prime objective of a microbe is to grow more, and this is quite feasible in the microbe. But if you go to the mammalian or multicellular organism, biomass maximization may not be very accurate. So since our workshop topic stick to microbe, where microbes are engineered to produce chemical or fuel, we think that the FBAs will be fine, and nobody will question on that. So, so to do the annotation and understand the number of reaction on the genome, then you go through a database, for example, Crick database, Bioseq, UniProt, big database, Brenda. So K-base is one of the most popular right now. K-base can give you much more detail of different metabolic pathway. You can also construct the draft metabolic model. which can also be curated for making the model much more accurate. For example, the model seed and K-base, these are the two. So the model K-base uses model seed, and model seed has an automated technique to construct the metabolic model. Right now we have seen that many models have been constructed using model seed, and model seed gives you a draft metabolic model from where you can start the starting point to construct a metabolic model. So some of you can try model seed maybe in the workshop or K-base so that you can streamline your metabolic model much more quickly. So another important thing in the metabolic network is the Boolean relationship, that is the GPR association, that is gene-protein reaction. So this is the relation which need to be there for every reaction. So without this mapping, you won't be able to have an accurate metabolic network. So the mapping, you can see succinate dehydrogenase, which has this gene, and then this gene can form a protein. So you can see this protein is a multimeric protein that have each gene contribute to each subunit of this protein to form a single protein which catalyzes two reactions. So this kind of mapping you have to construct for each and every reaction. You can understand the amount of effort needed to construct the genome scale metabolic network. And the more accurate this mapping, your model is also become more accurate. So for Saccharomyces cerevisiae and E. coli, most accurate model is available for E. coli, and then Saccharomyces cerevisiae and so on. So most of the models need to be validated by knocking a gene to see what is the phenotype, right? So the genes which are associated with the growth, you can easily compare, but the genes which are not associated with growth, they are very difficult to compare. But still, you know how many essential genes are present when you compare the in silico model and the experimental data. So the overall the summary of the reconstruction process where we annotate the genome and then find out the number of reactions, and then you also balance the charge and mass of each and every reaction, and using the set of all reactions, you try to construct a biomass equation. So biomass equation, you can see the biomass coefficient for the amino acid, lipid, RNA, DNA, pigment, if you are using algae or cyanobacteria. So this all the components which are present in the cellular biomass that you can also characterize experimentally by drying the cell and then and measuring each of the components that will comprise this biomass equation. And the biomass equation is experimentally measured while you make the metabolic network. And then you go for the then you build this stoichiometric matrix, and once you have the stoichiometric matrix, you can solve this equation to get the metabolic fluxes. So the metabolic fluxes, so you apply the steady state condition, that is S dot V equal to 0, and when you apply S dot V equal to 0, that is the S matrix, you can also mathematically you can represent a null space. S dot V is nothing but the null space, that is all the solution of the allowed fluxes will lie inside this space shown in gray color. So that is, this space is known as null space. So the null space you can easily calculate for a given matrix by just by taking a dot product equal to 0. That is S dot B equal to 0. And then you apply different regulatory constants. So we will also show you how you can apply different regulatory constants. Regulatory context nowadays applies in terms of RNA-seq data or expression data, where you constrain your metabolic network. Here you can see that your the null space is much more wide, but there is an optimal solution which is shown over here that lies at the edge of the this polyhedra cone, and this is the point where your biomass is maximum. So your fluxes will lie everywhere, but only one point you know that your biomass is maximum, and this is the idea of flux balance analysis. The flux balance analysis says that you maximize the biomass so that your fluxes are feasible, which is very much compared to the experimental condition. And this is only possible because you are actually maximizing the biomass, and there is only one optimal point. out of many allowed states, that many of the states are eliminated by regulation. So, the regulatory constraints are also important to constrain your metabolic network. Once you constrain the metabolic network, then your solution space will reduce and your solution will lie on a single point where your biomass is maximum. So this is good for the micro, but when you go to mammalian or multicellular organisms, then biomass maximization is not actually much more promising to predict the fluxes. So this the integration of multi-omics data is another emerging field. How we can integrate the gene expression data or proteomics data or metabolomics data into your genome cell model, so that your model will become a context-specific model. So you can actually build the model much more constrained. You apply as many constraints as much possible based on available data you have with you to predict metabolic fluxes. So this way you can really get much more accurate fluxes by applying different constraints, which can be, for example, all the RNA-seq data which are readily available today, which you can apply into this model to make the model context specific. So to summarize the flux balance analysis, you can write the mass balance equation, that is dC by dt equal to S11 dot V1. So you can see that when you write the mass balance equation, that is the time derivative of the concentration as a function of fluxes. So V1, V2, V3 up to Vn, that is the number of reactions you have in the metabolic network, and this you associate with a variable V1, V2 so that you want to calculate. And this time derivative of the concentration you can write for each and every metabolite to write these equations. So each equation you can and then ultimately you put S dot B equal to 0. That is the steady state approximation. So this is the approximation which allows you to calculate the metabolic fluxes. We assume that there is no metabolite accumulation inside the cell. The cell actually during the steady state condition, that when the cell is growing in the exponential phase, we are saying that there is no accumulation of the metabolite, and that's why you could write S dot V equal to 0. So the flux balance problem statement, which can be written as maximization of the V biomass, that is the V biomass is nothing but the growth rate, and that's subjected to these steady state condition, that is S dot V equal to 0, and then the boundary condition, that is so the boundary condition gives you an idea that whether the reaction is reversible or not, or a irreversible reaction. Based on that, you have some delta G value which are available for many of the reactions that you can put as a constant to get much more accurate fluxes. So using this technique, we have applied for reconstruction of a non-conventional yeast, that is Lakensia fluberii, while we build the metabolic model for this organism using already existing model for yeast. So yeast, as you know, there are a few more metabolic models which are available. One of the most well-known is Saccharomyces cerevisiae model, and then we have many other models which are available today. Most of the yeast metabolic models nowadays are available. So you make use of those existing models to build the metabolic model for Lakensia fluberii. So which comprises of 1235 reactions, and then we have the 1179 metabolites, 730 genes, and so on. So it is distributed in 8 components for building this model. And then we validated using the experimental data. So the experimental data, the knockout data we have compared. So two different media has been considered, the rich media and the minimal media. So in the rich media, you can see that the false positives are very less, like 90, and then when we shift into minimal media, then the number of false positives increased, and that is the main reason behind, because Lakensia fluberii has a less number of duplicate genes compared to Saccharomyces cerevisiae. That's why when you move from minimal rich media to minimal media, then the number of false positives increases. And then we also constructed the context-specific metabolic model for Lakensia fluberii for understanding the uracil degradation pathway, Luckily we have some expression data for this strain L. blueberry. So we use those expression data to make the context-specific model which grows on uracil, and we try to understand which are the metabolic pathway got affected because of uracil degradation, and we found that purine metabolism, histidine metabolism. So many of the metabolism is we got from differential flux analysis to understand which of the pathways got affected because of this uracil degradation. So this gives an idea of how many pathways got affected after constructing this context-specific model, and then compared with the original model to understand how different pathways are affected. So using this concept, we also build the metabolic model for lung cell which are infected by SARS-CoV-2. So we build the metabolic model for human lung cell in the normal condition. NHB is the normal cell, and then we have the SARS-CoV-2 infected lung cell. Those metabolic network have been constructed. You can see the number of reactions, 7,270 for the normal cell, and then with the infected cell we have 7,255 reactions. And then the biopsy sample were also available. So this context-specific model, as I told you, it uses the expression data. So the expression, the RNA-seq data was available for different patients who and the biopsy sample, the tissue-specific sample which I collected from a person who died in SARS-CoV-2, their lungs tissue were collected, and RNA-seq data was available, and we use those data to build the metabolic model for lung-specific tissue and the COVID-infected lung tissue. Then after constructing the metabolic network, then next is to do the flux analysis. The flux analysis also reveal some of the metabolic pathway which are already known to be dark target. For example, we have seen the fatty acid synthesis has increased in SARS-CoV-2, whereas degradation of fatty acid like beta oxidation pathway has downregulated based on the differential flux analysis. And also we have seen that anorthoside acid synthesis to sphenoid metabolism has also upregulated. So this metabolic pathway got upregulated because they have the inflammatory molecule which are a quick response to the virus, the immune response of a human body to the virus. Because of that, this pathway is upregulated. And another pathway we saw that got affected, that is the cholesterol biosynthesis. So the K-Russell pathway got downregulated, or whereas Block pathway got upregulated. K-Russell pathway is the precursor molecule for vitamin C synthesis. As you know that during the COVID time, the people started taking vitamin C because the vitamin C production got reduced because of the downregulation of the K-Russell pathway. So some of the metabolic pathways got upregulated, downregulated, have been compared, and we found there is a good comparison with the experimental finding. Apart from that, the genome scale metabolic model are also used today to construct the synthetic consortia, synthetic microbial consortia. So in metabolic modeling, which we already discussed, was on the single cell level. So suppose you want to construct a community, so then you combine several of these metabolic model into. So here you can see a co-culture system was developed where you combine two microbes in this culture. So now we want to understand the biology, that is the interaction of these two microorganisms. Then you have to build a community modeling. So we are working on this community metabolic modeling to understand how these two microbes are interacting, or you can go into many microbe systems to understand their biology. So for using this concept, we are using the termite gut microbiome to understand how different microbes are interacting. First of all, in the gut microbiome, we have thousands of microorganisms. So which one to select, which one not to select for community metabolic modeling? For that, we do the metagenomic analysis. So we construct a metabolic interaction network in the gut microbiome. So this kind of gut microbiome network, metabolic interaction network, we are working for termite, bovine, human gut to understand their interaction, and then finally constructing the metabolic network. So this is a species-wide metabolic interaction network in coniger as a type of termite. Within the gut, we know that termite guts are involved in degradation of lignocellulosic biomass. So we want to understand that what are different microbes which are essential for degradation of lignocellulosic biomass. For that, we construct a metabolic interaction network, and some of the crucial microbes which are involved in degradation of the lignocellulosic biomass was identified in this method. So this apart from this genome scale metabolic model, community metabolic model, then another way you can do the metabolic flux analysis is basically 13C method. This is much more experimental method. As I told you initially, that genome scale metabolic modeling and FBA doesn't require experimental data. You can incorporate experimental data, but an experiment is not mandatory. But in 13C method, you have to perform the experiment, and the experiment that you have to do is basically growing the cell in 13C level substrate. And then after you have grown the cell in 13C level substrate, then you collect the cell and lyse the cell and take out the metabolite and measure in a GC mass or LC mass. And using this mass spectrometry, you will be able to calculate the metabolite leveling pattern. And this metabolic leveling pattern can be used to constrain your metabolic pathway. Earlier I told you that you can constrain the metabolic network using the regulatory constraint, but also metabolic leveling is used in 13C method to constrain the metabolic network so that your fluxes are much more accurate. So you can have a high resolved metabolic fluxes. So combining the data with metabolic model, here also you need a metabolic model, but a reduced metabolic model. So 13C method is generally done on a reduced network because these calculations are much more computationally expensive. So to reduce the calculation, and also we generally construct a smaller network. So in this method, what we do is allow the cell to grow in a 13C level substrate, that is your first carbon is level. So you can see that in the glucose, this first carbon is C13 level, and then you allow the cell to grow on the level glucose. After some time, what will happen that most of your metabolites will also be labeled using 13C. And when it is in the exponential phase, and then what do? You collect the sample, and you collect a huge amount of sample so that you can do the GC mass or the metabolite measurement. Using GC mass can be done without any problem. So you collect the sample in a tube and then go through a GC mass analysis. What you get is basically a profile. It says that the fraction of the molecule which are first carbon level, second carbon level, 3 carbon level, like that you have a profile. So 0 means none of the carbon are level. So this profile you can generally get for amino acid, but you can measure metabolite level for in fact all the metabolite. So you can use as many metabolite in your study to constrain your metabolic network, which can give you much more accurate metabolic fluxes. So these are the amino acid leveling pattern which are obtained from experiment, and those are used to constrain the metabolic network to get much more accurate flux. The fluxes on the right hand side, you can see that the fluxes we get from the experiment. So that's why sometimes they are called experimentally derived metabolic fluxes, and which are much more accurate compared to FD. So the algorithm which gives you the metabolic fluxes, that is, we choose the flux distribution and then predict the amino acid leveling using the flux distribution, and then you compare with the experimental data. So the prediction of amino acid leveling, you start with a distribution of fluxes, so it can be random, and then you compare with the experimental data, that means the leveling you got from the GC mass. So on one hand you calculate the metabolite leveling using the flux distribution and then compare with the experimental data to get the much more accurate fluxes. So some of the equations which are used to calculate the metabolic fluxes, that means you have to compare the leveling of the metabolite. For that you use error function. So here you don't maximize the biomass, but what you do, you minimize the error function. So the error function is basically the mass distribution vector, that is the leveling pattern you get from the DC mass, that is through experiment, and the leveling pattern you got from the flux distribution computation. So on one hand you have the computational value and the experimental value, then you take a difference, and when the difference is very very small, like 10 to the power minus 20 or even smaller, then you stop your calculation. That is the recursive method where I keep on calculating. That's why 13C NFA must sometimes become very computationally expensive because of this comparison. So unless you get a very good comparison, the calculation will keep on going and it will take a long time. So this is one of the results which we have shown here, how we can compare the leveling pattern of experiment and computation. So the red one is basically the experimental leveling pattern, and the blue ones are basically the computational leveling pattern. So you can see that when the fitting is very good, then your fluxes are also accurate. So this is the concept which is used in 13C NFA. You feed the leveling pattern to get the much more accurate metabolic fluxes. And this is done basically using the atom-atom transition map. So the atom-atom transition map which is shown over here, like acetyl pyruvate giving you acetyl coenzyme. Basically pyruvate has 3 carbon, and then 3 carbon is divided into 2 carbon in acetyl coenzyme and 1 carbon in the carbon. And you should know which carbon goes where. So you can see that the last 2 carbon goes into acetyl coenzyme and the first carbon goes into carbon dioxide. So because we know this mapping experimentally, that's why we could do the metabolic fluxes for using 13C NFA. So using this atom-atom transition, we can calculate the mass distribution vector for acetyl coenzyme by knowing the mass distribution vector for pyruvate and acetaldehyde. So if you know the mass distribution vector of pyruvate and acetaldehyde, one can calculate the mass distribution vector of acetyl coenzyme provided I know the flux distribution. So the flux distribution you can see are used to normalize this distribution vector, so that you get the distribution vector of acetyl coenzyme. So given a value of mass distribution vector of pyruvate and acetaldehyde, one can get the mass distribution vector of acetyl coenzyme by normalizing this flux level. So the metabolic flux analysis, which I have already discussed, the flux balance analysis and the 13C NFA. The flux balance analysis uses linear programming, and whereas 13C NFA uses a nonlinear programming, whereas FBA uses a genome scale model, whereas 13C NFA is a simplified model or a reduced network. And the equations which are involved in the FBA and 13C NFA are shown over here. The FBA which involves the maximization problem of biomass subject to steady state condition and boundary condition, whereas in 13C NFA we minimize the mass distribution vector, that is the leveling pattern you get from experiment and as well as from the simulation, and this difference you minimize subject to steady state condition. Here you apply the isotopic steady state condition, and then you calculate the mass distribution vector one by one, just I have shown in the previous slide. So you calculate mass distribution vector for each of the metabolite inside the cell. So this calculation is much more computationally expensive. That's why you make the network very small, so that your calculation goes quickly. So these are the two flux analysis tools available. You use the metabolic steady state, and whereas in 13C NFA you use a metabolic and isotopic steady state. So you can understand that 13C NFA works on a reduced network, where FBA works in a genome scale network. So genome scale network was much more efficient because you can do cofactor balancing, you can do a lot of understanding in terms of how fluxes are moving from one compartment to the other compartment. But in the 13C NFA we have a reduced network, so this can be overcome by combining these two methods. So 2-scale 13C NFA, which was developed earlier, were combined two different methods. That one is the FBA and the other is basically the 13C NFA. So how you can combine these two methods? So the combination is basically, so you want to use a genome scale stoichiometry rather than central carbon stoichiometry, which is used in 13C NFA. So you can make use of the genome scale model. Also you can make use of the extracellular flux measurement and 13C leveling data. So if you combine these three things, then it becomes 2-scale 13C NFA, because you want to do 13C NFA at the genome scale level, so that you can do cofactor balancing and other study you can do. So this method which take care of this patency NFA at the genome scale level, where you can see that the central metabolic networks are solved at a higher resolution using nonlinear equation, whereas the remaining part of the network are solved using normal FB, that is metabolite balances. So at a two different resolution, you can actually calculate the fluxes. So the central metabolic network use the carbon leveling tracking simulation, where you use the nonlinear equation, whereas in the rest part, the remaining part of the network you use the flux balance analysis. So this way you can combine these two methods to calculate the metabolic fluxes for the entire network at the genome scale level, which can include the genome scale flux data for further analysis. So using that, the genome scale metabolic flux analysis have been done for L. Klubery, and then we have also calculated the metabolic fluxes for the model we have developed for Lacancia Klubery to know how the carbons are actually flowing at different part of the network. And also you can do this kind of cofactor balance, as I told, that how much amount of acetyl coenzyme is formed and then how many reactions the acetyl coenzyme is consumed. So this kind of figure you can or understanding you can have when you have it through scale patency NFA. Then so far we discussed about the flux analysis tool, different flux analysis tool which are applied. Now I will go through the engineering part, the metabolic engineering part. So the engineering that you can do using genome engineering tool, for example, CRISPR-Cas9 or any other method which are developed for yeast. So using the CRISPR-Cas system, you can design a 20 nucleotide sequence that you want to target. So using Cas9, you can delete the chromosome at a particular location given by this 20 nucleotide, and then you try to repair the cell. So in the metabolic engineering, we generally don't allow the cell to repair on its own. So the repairs are done using generally when you target the chromosome, the cell will repair on its own, but you don't want this repair rather than you use a homologous directed repair. So HDR is used in the double standard break, where you can knock in, knock out any gene. So this is because in metabolic engineering you always delete a gene or integrate a gene or incorporate a mutation. So whenever the CRISPR-Cas9 break the chromosome and then you insert the gene of your interest, by the technique which we use is basically homologous repair, homologous directed repair that helps you to knock out, knock in and knock down. So the CRISPR-Cas9 tool were developed for many of the yeast, for example, Saccharomyces cerevisiae, Pichia pastoris, Variolae pelotica, K. marcinias, and Candida albicans. So some of the techniques you can use to actually engineer the yeast for production of biofuel. So this is taken from NEB, where you can see that you can do genome engineering, where you can clip the double stranded DNA and then you can design your donor DNA in such a way that it flanks along the cleavage, so that a gene can be integrated. Similarly, you can integrate activators such that you can downregulate the gene and so on. So these are the strategies we use for like the disruption, deletion, integration for building metabolic pathway. So lastly, I'll give one or two examples how you can use the flux analysis and genome engineering tool for production of fatty acid. So the fatty acid production, for example, in Saccharomyces cerevisiae, which we targeted by upregulating SEC FAS1, FAS2, which gives you fatty acid. Then we also deleted the pathway that allow the fatty acid coenzyme to go to come in a reverse direction, and you use a TSA for production of free fatty acids. So free fatty acid production gives you an idea like how much improvement you are doing in terms of fatty acid production, and that we estimated using 13C-NFA. So using to improve the production much more efficiently, what we have done, first we have upregulated FAS1, FAS2, SEC1, and also deleted FA1, FA4 for production of free fatty acid. And then we have done the metabolic flux analysis to understand how the carbons are flowing in different metabolic pathways. So the acetyl coenzyme production and consumption has been calculated using 13C-NFA, and then we understood that the fluxes of the acetyl coenzyme is a precursor molecule for fatty acid biosynthesis. The more the acetyl coenzyme production, the better is the fatty acid production. So for that we understood how acetyl coenzyme is used by different reactions, and then we found that the acetyl coenzyme for exponential phase and respiratory phase have been compared, and we have seen the malate synthesis, which consumes a lot of acetyl coenzyme in respiratory phase. You can see that the fluxes are more in the respiratory phase in the malate synthesis pathway and compared to the exponential phase. So then what we have done based on this flux prediction, what we have done is basically downregulate the malate synthesis pathway and then we can improve the production of free fatty acids. So we got an improvement in free fatty acids from 460 milligram per liter to 780 milligram per liter just by downregulating the malate synthesis and also expressing the ATP citrate lyase, which convert the citrate from mitochondria to acetyl coenzyme. So these two modifications has improved a lot for the production of free fatty acid. Another molecule we have targeted the fatty alcohol production in Saccharomyces cerevisiae, and this, the fatty alcohol production has improved from 1.2 gram per liter in flask and 6 gram per liter in cage batch by genetic modification that we targeted for this Saccharomyces cerevisiae. So for that we integrated the Pha gene for fatty alcohol production from mass muscular. You can see that the expression of different Pha which has been collected for only mass muscular, you can see that the fatty alcohol production is much more higher. So the Pha was taken from mass muscular, and also we did the metabolic flux analysis to know which are the pathways which consume NADH. NADPH is one of the molecules which is used for fatty acid biosynthesis. So there are some competing pathways which consume the NADPH. So those pathways are deleted. So for example, we have deleted GDH1, and then also we have deleted the HFDE1 and ADAC. These are competing pathways taking the carbon away from fatty alcohol production. And also we integrated the Pha gene at different locus. So the entire engineering was done at the genome engineering tools were used, for example, CRISPR-Cas9 to integrate this, to do this intervention. So you can see there are many deletion, many integration were done. This is only possible because of the CRISPR-Cas9, because in using CRISPR-Cas9 we can do any number of genetic intervention without leaving any mark. So this starting with almost zero fatty alcohol production, and we got around 80 milligram per liter of fatty alcohol production by doing this deletion and integration. And so finally, in summary, what we learned today is basically genome scale metabolic modeling, how you can do genome scale modeling for non-conventional yeast, for example, L. blueberry, and then then you can do the 13C MFA for flux analysis to calculate the metabolic fluxes, and then using the flux data, how you can predict or target the gene for genetic intervention for improvement of free fatty acid production or fatty alcohol production based on flux data. And also we discuss about the CRISPR-Cas9 technology, which is used in yeast for integration of different metabolic pathway, and or we want to delete a gene or integrate a gene using this genome engineering tool. So we have this systems on synthetic biology group in IIT Kharagpur. We work on genome scale modeling and metabolic engineering. So many of them initially who joined has left the group, and they joined. For example, Bharat graduated with PhD and went to Columbia University. Piyush went to Harvard University, and many new people have joined during the COVID time, so they have started working on it. So hope we'll get new results very soon. Thank you. I will take a few questions.
All right, Dr. Kosh, thank you very much for the enlightening talk covering both the basics of metabolic flux analysis as well as flux balance analysis, and also genome scale metabolic flux analysis, and supplemented all this theoretical information with actual experimental evidence of these applications in the yeast models using CRISPR-Cas based tools for production of fatty acids as well as fatty alcohol. Thank you very much for the enlightening talk. We are now open for the questions. Again, we can ask the online audience first if they have any questions. They can raise their hand and then we can ask the questions. Do we have any questions from the offline audience? Yes, we do have.
Hello sir, thank you for the wonderful talk. My name is Lokesh. So my question is that, the both techniques, flux balance analysis and metabolic flux analysis, you said we do it at steady state, but in what we do is that in cyanobacteria, we do the metabolic flux analysis in unsteady, isotopic unsteady state. So what are your thoughts in overcoming that challenge? I mean, I want to do it at unsteady state. So what are your thoughts on that?
Yes, I have not discussed about the non-stationary 13C. So the organism, for example, for cyanobacteria or any other photosynthetic organism, there you will not be able to reach to the steady state so quickly. You understand that 13C NFA is very costly experiment. So the isotope level glucose, as you know, it comes very expensive. So if you have to wait for longer time, then your expense will increase, because you have to wait for the steady state, because flux balance analysis or the normal 13C NFA only work on a steady state condition, that is H dot equal to 0. But if you want to work on an organism which doesn't reach to steady state so quickly, then you have to wait for a longer time, then you cannot apply this method. So what you use basically a non-stationary 13C NFA, where you solve the differential equation which is much more different in the sense that where you have to solve those equations. That dc by dt we have written, right? dc by dt becomes 0. That is, the concentration of the metabolite becomes 0 with that approximation. That approximation doesn't work here. So this one basically. Yes. So dc by dt is equal to s dot v, which dc by dt is no longer 0 for non-stationary 13 scenario. That means you cannot apply steady state condition. S dot v is not 0. So this for that you actually have to solve the differential equation. So the for photosynthetic organism that require this solving this equation. You have a software right now, INCA, which is developed by Jamie Young. You can actually get an academic license for those kind of flux analysis. I hope I'm able to answer your question.
Yes, sir. Thank you.
Any other question?
Shirish, I can see online Alexandra want to ask question.
May I?
Yeah, sure. Absolutely.
Thanks. Thank you. Thank you for the very nice talk. I'm Alexandra Djukic-Vukovic. My question is partly related to the previous one. I mean, these databases and these softwares you were mentioning, are they free, or are some of them free, or what are the conditions for licensing? I mean, academic licensing or anything like some advice from you as an expert?
Thank you. You have a very interesting question. As you know, the model seed actually funded by DOE in USA. So most of the database which you can see over here are funded by the government, so they are free. So that is the benefit of the research community. For example, KBase is DOE funded, and it is freely available. So you can create a login and start building the model. So I actually use KBase for building the model for yeast. But if you want to build the model for bacteria which doesn't have any compartment, then you can use model seed. And model seed can be used in any model initially, because you need a starting point. For example, if you want to build the metabolic model for cyanobacteria, you can build the metabolic model using the model seed initially, but model seed will give you the metabolic model where it will lack the photosynthetic pathway that you have to add manually. So model building is partly automated and partly manual. So manually you have to curate the model so that it becomes accurate. But if it is a bacteria, then the curation is much more easier, but much more complex organisms like photosynthetic that require much more effort from your side. But you can start with model seed or KBase to have a draft model. The starting model is much more important. And so once you have something to start with, then you can build on that based on the data you have in database. For example, cake database, Uniport, Biosig. But model seed and KBase are actually help you to begin from a scratch. So you don't know absolutely about that organism. You use those platform to start your model building.
Thank you very much. Yeah, thank you very much.
Thank you. Thank you.
Do we have any other questions? All right. So Rakshit has asked, does it mean FBA is only applicable for the exponential growth phase of the organism?
FBA, so initially I told you that FBA actually deal with steady state condition and also biomass maximization. So that you cannot apply when it comes to mammalian cell. So we build the metabolic model for lung cell infected with SARS-CoV-2. We do we estimate the metabolic flux through sampling procedure, flux sampling. So some of the steady state condition and which are the approximation for microbial cell cannot be used in another type of a cell. So steady state condition is just an approximation that may not be true for all the microorganisms. For example, photosynthetic organism you cannot apply steady state condition because you have to wait. You know that photosynthetic organism takes longer time to it will take much more time to go to steady state condition. That's why you cannot apply much more accurately. Hope I answered your question. Right?
Yes. So I guess let's thank Dr. Kosh for taking time this Saturday morning to share his insights with us, and again with the interaction that we have seen, I think the talk was very well received and appreciated. Thank you, Dr. Kosh, for your interaction today.
Thank you, Dr. Srivastava. I really appreciate for organizing this workshop. Really interesting. I also got a nice interaction with others. Hope we keep on organizing this kind of workshop in future as well to help the community, which will enhance our interaction in great detail.
Yeah.
Thank you.
And actually, we also need to thank DPT for funding these workshops at ICGV, and we do have a series of workshops on different topics, and thankfully this one where we are focusing on bioenergy and flux analysis, synthetic biology applications in bioenergy, was one of the workshops that was selected for this series of events. So, yeah.
Thank you so much for the invitation.
Thank you, Dr. Ghosh. Thank you very much. It was pleasant.
Thank you.
So for the for the online participants, we will have the hands-on session, which, as I mentioned, we will have it shared over the Zoom. So you guys are welcome, and we'll be joining back again in like 5-10 minutes. We have to change rooms. So once we reach there, you can either join back in 5-10 minutes, and then we can 5 minutes actually, and then we can start our hands-on session on genome scale metabolic model analysis.